Hello, this is Nerz-Stormes. I'm in a place that may be familiar to some of my students, my dining room. I'm going to try to give a very quick review of the type of research that I've been attempting to do at Bryn Mawr College. I must say I have a very fledgling research program given my teaching load, but I am trying to do a couple of projects. Through my own personal experience, I became very interested in medicinal compounds, particularly compounds that are active against cancer. And among those compounds, I am particularly interested in drugs that can cross the brain barrier and also those that cannot cross the brain barrier at all. The blood-brain barrier is something that we have that keeps toxins really out of our brain. So we evolved in such a way that we have really what really consists of some very tightly packed cells around the blood vessels of our brain that have some active transport systems built into them that keep toxins from entering our brain, and so this really protects us. But on the other hand, many chemotherapeutic agents and many agents, even biological agents, that might be active against brain tumors and brain metastases just simply can't get into the brain. So I was very interested in this, and so I actually studied all the compounds that are used in treating cancer, and I tried to assess which compounds had data that indicated they do enter the brain or cross the brain barrier, and those that absolutely did not, and then those where I just really couldn't tell. And again, basing this on data, not modeling. And in this assessment, I became very intrigued by this particular compound, which is called verinostat. And verinostat, to me, was such a simple, tiny little compound. Now, the reason I became sort of interested in it is it's a new compound. It's a drug that's just being marketed now, and it's known to cross the blood-brain barrier. The class of compound that it is, it's a compound that's known as a HiDAC inhibitor or a histone deacetylase inhibitor, and histone deacetylases are involved in some cancer pathways, including breast cancer. And this drug is used to treat a particular type of cancer right now. And I was really interested in it because I thought it was so simple, and there was so much known about it. It's known that this end of the molecule is involved when it's sitting in its enzyme pocket. When it's sitting in the enzyme pocket, this part of the molecule is bound to a metal. This part is kind of called like the linker part of the molecule, and this part is the recognition part, and this usually has to be an aromatic. This end has to have certain heteroatoms in order for it to bind the metal, or a certain organization of those atoms. So I thought this molecule was so simple, and I thought that's so interesting that it crosses the blood-brain barrier. And then scanning all the different HiDAC inhibitors that have been made, this molecule really jumped out at me. And the reason it really jumped out at me is because it had particularly high activity, and it looked like something that would be easy to make. So I thought it would be really interesting to make a whole series of compounds related to this molecule, MS-275. And I should mention something else about it. It does not cross the brain barrier, and this has been demonstrated in primates that this molecule does not cross the brain barrier. So I thought it would be interesting to make a series of these molecules. This is the recognition part, and you can very easily vary this section, the middle section, and the metal binding section. And I thought it would be interesting for students to make a whole series of these, and then to study maybe both by modeling and by looking at octanol-water ratios, whether the likelihood that the molecules we would make, the varied molecules, would cross the brain barrier. And also, another thing I discovered in my review of the literature was that most of the compounds that do cross the brain barrier and are active in brain cancers, primary brain cancers, and metastatic brain tumors, most of these compounds either do not bind albumin at all, or only partially bind albumin in the blood. Many pharmaceuticals are carried bound to albumin. And this may not sound surprising to people, but this was very surprising to me because there are many compounds that are bound 100% to albumin that do cross the brain barrier. So one of the things I thought would be interesting to study too is if we could make some sort of correlation between the compounds we were making, their ability to the extent to which they bind to albumin, and their octanol water partitioning, or there are programs you can use to sort of model their brain barrier, their potential brain barrier permeability. So that's one project, and that's one project we've started a little bit. The other thing I'm really interested in are platelets, and this again is through personal experience. I personally have had trouble with my platelets. It's called thrombocytopenia, and it's due to cancer treatments. And interestingly, a student here came up to me and said, "Dr. Nerz, in Bangladesh, papaya leaf extract is used to raise people's platelets when they have dengue fever." And I'm very skeptical about these kind of anecdotal cures, very, very skeptical. So I started to do a lot of reading, but I continued to have the problem, and it was actually interfering with my own treatment. So I decided to just start taking it, and I ordered it from Amazon.com, and amazingly, it raised my platelets tremendously and has maintained my platelets at a very, very high level and enabled me to continue my therapy. So I got very, very, very intrigued by this, and I kept reading and reading and reading and reading, and found out there was almost nothing known about this. So one of the things I really want to start doing this year is I want to take this extract, and and a little bit of work has been done on this, but I want to actually extract the components, and in particular I'm interested in the components that are less than 1,000 molecular weight. And I want to find out exactly what's in there, how much is in there, and try to figure out which component or components are responsible for this activity. It is actually true that there is no current treatment. There's something on the horizon called Promacta, but there's no current treatment for thrombocytopenia, and this is a very, very, very simple solution to the problem. Of course, I am only a sample of one, and perhaps my experience means nothing, but there's actually many, there's a lot of information out there that states that this may be true, and I actually found a study that showed that the papaya leaf extract does raise the platelets in mice. So I'm kind of interested in exploring that too, and I thought that would be something students might be interested in. Okay, thank you.